So this video is about viruses and the introduction to viruses. So in the self-study, you're instructed to read the first chapter of Murray's Medical Microbiology. And in that chapter, they go through basically the 4 types of organisms that make humans sick. So there's bacteria, there's viruses, which we're going to talk about here, there's parasites, and there's fungi. So when we think about these 4 organisms, we also need to think about what makes them different because they're likely to cause different forms of disease. And we're going to start with viruses. So viruses luckily come with 3 unique features. The first one is that they are not alive. Well, not technically. This means that they are obligate intracellular organisms. So what do I actually mean by that? I actually mean that in a world full of parasites and microorganisms, in my opinion, viruses are the real parasites because they literally are not alive anytime they are outside of another cell. They require all of the machinery and components for replication and for their very existence. So they're an obligate intracellular organism. If they're not inside another cell, they're not replicating, they're not doing anything. The second important thing to note about viruses, they're really, really small. What does this mean? Well, viruses were originally referred to as filterable agents. That basically means that filters that were designed to catch other microorganisms didn't catch viruses. So that should tell you something about their size. Other than that, it's pretty self-explanatory. The last thing is that they cause pathology by multiplication. So when we think about things like bacteria and parasites, they might produce a toxin that can cause damage, cause disease. Viruses do not produce any sort of toxin. They cause disease just by existing, which if you think about it is pretty genius. So how do they do this? Well, one is multiplication. Their very multiplication causes changes to our DNA because their genome can actually get substituted for pieces of our genome. And we see that in some forms of cancer that exist that have a viral etiology. The other thing that they might do is they lyse cells. So when a virus replicates, eventually it needs to get out of the host cell so it can go attack another host cell. And one of the ways it does that is by lysing cells, which, big shock, is bad for the host, the host who has the cells, namely us. Okay, let's talk about the physical structure of viruses now. The first thing we have to get clear is a little bit of nomenclature. So a virus is actually a group of virions, which are the individual viral particles, much the way mankind is a group of individual humans. Make sense? Okay. So a virus is really a very simple machine when you get down to its core. A virus, a single virion, can be made of what is basically called a nucleocapsid. A nucleocapsid is literally just the genome, which is going to be either DNA or RNA, never both, and some structural proteins. The structural proteins will basically provide a hardy shell that protects the genome of the virion. So you can see that here in this image. So you've got DNA or RNA plus the structural proteins, basically gets you what's called a nucleocapsid. If that's the only thing that makes up the virion, it's called a naked capsid virus. Whenever we think about naked capsid viruses, it's a good time to talk about viral proteins. So there are kind of 2 forms of viral proteins. There's your structural proteins and your non-structural proteins. Structural proteins are going to take on one of 2 conformations. They're going to be helical or icosahedral, and that's going to form your capsid. The capsid is going to complex with the genome, and that's going to form, you guessed it, your nucleocapsid, which is technically a functional virus. In fact, these naked capsid viruses make up your gastrointestinal tract viruses. So anytime you've gotten a viral gastroenteritis, it's because of one of these naked capsid viruses, this really simple machine. There are also non-structural proteins that can aid in attachment or release from the cell. The other way we can see viruses show up instead of just a naked capsid virus is an enveloped virus. So enveloped viruses are basically you took a naked capsid virus and you wrapped it in a host membrane shell. So you wrapped it in host membrane, basically. So this is just a glycolipid membrane, just like we have, that is wrapped around a nucleocapsid. And often this membrane will contain various proteins, and these would be non-structural proteins. These proteins will include things like the viral attachment proteins. Viral attachment proteins are pretty important. They often look like spikes, as you can see here, and some of these glycoproteins are what bind to cells and help the virus eventually enter the cell. An important one to keep aware of is hemagglutinin. Hemagglutinin literally is a viral attachment protein that allows viruses to bind to erythrocytes or red blood cells. Some glycoproteins, though, have other functions. So there's also the necessity for the virus to leave the cell. So once it's finished replicating, how does it get out? And they'll have viral proteins for that as well. So for instance, in flu, there's a protein known as neuraminidase, which allows the virus to leave the cell. It's necessary for it to actually get out once it's been replicated. These glycoproteins, especially the viral attachment proteins, often elicit immune responses and are often a target of antiviral drugs. So let's talk a little bit over here about the viral genome. So I already mentioned that it can be RNA or DNA, but never both. Viral genomes can come in a variety of formats. They can be single or double-stranded. Single-stranded genomes may be positive or negative sense, and double-stranded viral genomes will be ambisense, containing both plus and minus regions. The RNA genome can actually be segmented into pieces, with each piece encoding one or more genes. Viral genomes can be linear, or they can be circular. Specifically, DNA viral genomes can be circular and take on kind of a different conformation. So there are a whole bunch of viruses, so we needed a way to classify them, and there's kind of a variety of ways. So viruses can be classified on their size, because even though they're all really small, they still have a differential in how big or small they might be in comparison to other viruses. So if we look at just kind of some of our viral names down here, there are things like the picornaviruses. Picornaviruses are some of the smallest viruses that we're going to encounter. You can also take a look at them based on their morphology. Well, what do I mean by that? Well, morphology literally means, are they a naked capsid virus or are they an enveloped virus? So you can see here with the DNA viruses that they've broken this classification down into enveloped viruses, which includes poxvirus, herpes virus, many of you have probably heard of that, and hepadnaviruses. Naked viruses would be things like polyoma or parvoviruses, papillomaviruses, adenoviruses, and polyomaviruses. You can also think about them based on their mode of replication. Are they an RNA virus or a DNA virus? And then you can also think about the diseases they cause. Do they cause the common cold or do they cause hemorrhagic fever? Other ways of thinking of viruses include the animals they infect and the tissues and organs that they infect and their means of being transmitted. Are they transmitted via the fecal-oral route or are they transmitted in respiratory droplets? And then they're broken into families, and each of these families have their own suffix. So for instance, the coronavirus is a Coronaviridae family, but in general, we're just referring to them as a virus. So I would say coronavirus. Okay, so the major steps in viral replication are kind of the same for all viruses. The cell acts as a factory, providing substrates, energy, and replication or assembly machinery for the viral proteins and genomes. Any process that the virus requires that the host cell doesn't normally do must be encoded for by the virus. And this image kind of shows how that factory works. And anything in red here is actually a target for an antiviral, basically something that we use to treat viral infections. So for instance, you've got these protease inhibitors. They basically inhibit the activity of proteases, which are necessary for the virus maybe to assemble or to get out of the cell. There's also antibodies that basically block the ability of those viral attachment proteins to attach to the cell. So this occurs over the course of 8 basic steps, and we'll go through each of these steps. Events in the replicative cycle have basically been followed using a synchronized infection. A synchronized infection would really only occur in a laboratory. And this is also known as a one-step growth curve, which I've outlined for you here, and it would happen in cultured cells. This will actually occur when you infect a culture of cells with a high multiplicity of infection, basically a high amount of virions that's going to result in an infection that's going to rapidly multiply within your culture pool. So there are 4 stages in a one-step growth curve: attachment, eclipse phase, synthesis, maturation, and release. So attachment is pretty self-explanatory. The virion attaches to the host cell. It's going to recognize an appropriate target cell, attach to the cell, and penetrate the plasma membrane. Because remember, these viruses do not replicate outside a host cell. Then you have what's known as the eclipse phase. During the eclipse phase, most of the virions you started with start to disappear. Why? Because they're penetrating the cell, so you can't see them anymore. But once inside the cell, they'll uncoat and begin the process of replicating their genome and producing their viral proteins. As the eclipse phase ends, you begin to see synthesis of new virions. So now that it has replicated its genome and its proteins, it's starting to put together its nucleocapsids and get ready to mature, which is basically how the virions begin to assemble within the infected cell and then be released out into the extracellular space so that they can go on and infect another cell. So that's kind of the basic broad strokes view of viral replication. So let's take a closer look at each step of viral replication. So as I mentioned earlier, you've got recognition and attachment. In this stage, the virus binds to a specific plasma membrane receptor on host cells. This is typically accomplished through a viral attachment protein, like a hemagglutinin, as mentioned before, that we can find in the influenza virus. Remember, this is a receptor-mediated endocytic process. The interactions between receptors on the host cell and the virus are what provide both species and tissue tropism. Tropism literally means what cell types is it going to infect? Is it going to infect nerve cells like rabies virus does, or is it going to infect epithelial cells? It'll also determine host range, so what animals can infect. There are some viruses like flu, which can infect both birds and humans. There are also viruses like the human immunodeficiency virus, which really only infects humans. So the next step is penetration. So most viruses are taken up in some sort of vesicle. This occurs when interactions between multiple VAPs and cellular receptors initiate this internalization of the virus. Endocytosis is a normal process used by the cell to uptake receptor-bound molecules like hormones and other things, but it's also used to take up viruses. Then we move into uncoating. So you took it up in this nice little package, but the virus needs to get out of there if it's ever going to replicate itself. So the nucleocapsid has to be delivered to the site of replication within the cell and the capsid or envelope removed. The genome of DNA viruses has to be delivered to the nucleus. The genome of RNA viruses, on the other hand, is generally replicated in the cytoplasm, with the exception of influenza. Influenza is kind of a special snowflake, so it's typically replicated in the nucleus, whereas all other RNA viruses are replicated in the cytoplasm. An enveloped virus might fuse its membrane with the cellular membrane and deliver the nucleocapsid or genome directly to the cytoplasm. And this fusion activity might occur with a viral attachment protein or another protein. For example, the hemagglutinin of influenza A binds to sialic acid receptors on target epithelial cells in an acidic environment, and that basically exposes the hemagglutinin hydrophobic residues of the envelope, allowing for fusion of the host cell membrane with its capsid and its uncoating. Okay, at this point we move into transcription. So the cell's machinery for transcription and mRNA processing is typically found in the nucleus, right? So regardless of the genome that a virus is carrying, plus sense or minus sense RNA, plus sense or minus sense DNA, segmented, whatever, all viruses have to get to the point where they make mRNA. Most DNA viruses will use the cell's own DNA-dependent RNA polymerase II, whereas for RNA viruses, they're going to need to come up with some alternative replicative mechanism in the cytoplasm, because typically this is only available in the nucleus. So once it's accomplished transcription, it's going to need to move on to protein synthesis, which I've shown here in this blue circle over here. Much like our own replication, viruses perform translation to synthesize proteins. They can do this following transcription once mRNA is made. All viruses depend on the host cell ribosomes, tRNA, and mechanisms of post-translational modification to produce their proteins. Many of these proteins will be necessary for the virus to replicate the genome in the next step. So that's what we're going to do next. So we make all of the proteins and machinery because, remember, for RNA viruses, they might need to make their own polymerase because they're not going into the nucleus to use the host cell's polymerase. So they make things first, then they replicate the genome. So the genome that was in that nucleocapsid is kind of like a template so that they can make themselves again. But it's a template that first has to become mRNA to make proteins. So at this point, we're going to replicate the viral genome. This allows the virus to actually make new virions eventually. Finally, once all of this is done, we're going to assemble. How are we going to do that? Well, remember, it's a fairly simple machine. You're just going to take that genome you made, you're going to complex it with the capsid. If you have any glycoproteins, they're going to complex with that too. Once you've got all of that complexed together, it's time to leave the cell. So you can either be pinched off, as is being shown here, or if enough of them build up, pop, you can just lyse the cell. Okay, and that is basically intro to viruses. So in conclusion, viruses are really small intracellular obligate organisms that require the machinery and energy of another cell to live. And they cause their damage by multiplication, not secretion of toxins or anything like a bacteria or a parasite might. They consist of an RNA or DNA genome, never both, and some structural proteins that make up the capsid, together forming a nucleocapsid. And that nucleocapsid may or may not have a viral envelope. The viral replication cycle has the same 8 steps, starting with attachment, ending with release or lysis, regardless of the genome. But where these steps take place, in the cytoplasm or in the nucleus, may differ depending on if you're dealing with an RNA or a DNA virus. That's all for now. Thanks.